Hi everyone. I'm Nonhle Mlotshwa, here to talk about my work on the individual phenotypic variability of the bronze bream population, an important target species for our recreational marine shore-based fishery. Can you hear me? Well, okay, great. Forward. So when we talk about the response of fish to climate change, we usually look at the response of populations, but it's the individuals within the population that experience the environment, and the individuals are not the same with regard to their physiological fitness. As physiology sets the limits of what fish can do, it determines how fast they can swim, how efficiently they feed, and whether they survive. In general, studies have reported physiology of fish at a population level, not taking into consideration individual variability that may exist within the population, which makes us wonder if the response and adaptations of all individuals within a population to environmental variability are the same. This, of course, is not the case. Within fish populations exist diverse physiological phenotypes that can aid in the resilience and persistence of fish against climate change. A phenotypic variability within individuals is a result of their physiological and cognitive components, which is then reflected in their capacity, in their behavioral capacity and performance. As such, individual variability drives collective behaviors and group functioning, which then affect the structure and dynamics of community dynamics and population activities. The environmental conditions encountered by the organisms may influence the individual variability and thus the collective behavior of a population, but the environmental conditions may also be a result of individual variability. And this may result in natural selection, which ultimately shapes individual phenotypic variability within populations. Individual variability within populations comes in handy when fish are faced with two of the most influential climate-mediated stressors, which are temperature and oxygen. These are thought to be the primary drivers of performance. And exposure of fish to temperatures and oxygen levels that are beyond their tolerance limit for prolonged periods or at a rapid rate may result in fish kills, loss of diversity, potential ecosystem imbalances, and this has a negative influence on fisheries. And given our unique South African ecosystem, which is surrounded by two major currents, we have the cold, nutrient-rich Benguela on the west coast and the warm, fast-flowing Agulhas current on the east coast of South Africa. Individuals within this coastline are exposed to highly variable and sometimes extreme conditions. Therefore, understanding how fish adapt to these differing thermal and environmental regimes is essential in order to predict their response and future distribution. Now, our study species is the bronze bream, species name Pachymetopon grande. It is endemic to southern Africa. It is highly resident, slow-growing, late maturing, and long-lived. It can live up to 40 years. It is one of the dominant species in the recreational and small-scale fisheries of the southern coast, contributing to food security in coastal communities. And because of its longevity and site fertility, it is a good indicator species for monitoring ecosystem health and exploitation. The catch rates of this species over the past 30 years have declined substantially, leading to the classification of this species by the IUCN as near threatened. So our study sought to better understand the effect of temperature on the performance and response of coastal fishery species by investigating the effect of thermal variability on the aerobic scope of the bronze bream and identifying phenotypic variability in the performance of the individuals. So this was done using intermittent flow respirometry, which measured the rate of oxygen consumption of the fish. So in partnership with the Rock and Surf Super Pro League, RASPO, the Department of Ichthyology and Fisheries Science in Rhodes University managed to record data collected by competitive recreational anglers in order to inform improvements in coastal fish conservation and management strategies. Now, RASPO takes place in the Western Cape and the Eastern Cape of South Africa. So catch rates are recorded from the Kai Mouth to False Bay. And from this data, or from these catch records, we're able to identify the hotspot of bronze bream, which is here highlighted in red. And hence the selection of our sampling site, which is 30 minutes away from here. So our sampling site was Port Alfred, East Beach, located in the Eastern Cape province of South Africa. So this region is surrounded by the Agulhas Current, which has dynamic oceanic conditions, significant eddies, and due to intensification of upwelling favorable southeasterly and easterly winds, there's been a cooling trend observed in Port Alfred. So this graph shows the in situ temperatures recorded that we obtained from SciAB's acoustic tracking array platform, ATAB. So it basically shows the thermal variability that occurs within this area. So data was recorded from 2015 to 2022. So temperatures here went up as high as 21.9 and as low as 8.5. And this is what we use to inform the test temperatures that we're going to use for our respirometry. And because this is the environmental conditions that the bronze bream exists under. So we collected 24 fish with a total length ranging from 309 to 515. 4 fish were used for the pilot trials to basically determine the temperatures that were the CT min and CT max. And then 20 fish were used for experimental trials. And we used 3 temperatures, which were 10, 18, and 22. And this was a repeated measures experiment. And to measure the rate of oxygen consumption, we put our bronze bream in respirometry chambers. And from here, we're able to get the standard and maximum metabolic rate. We then calculated the difference between the two to get the aerobic scope, which is the oxygen consumption required to perform important physiological and biological activities. And from the aerobic scope values, we were able to determine the different performance phenotypes of each fish, classifying them as either low or high or intermediate performers. And statistical analysis and data handling were done according to Bailey et al. 2025, where we used linear mixed effects models to model the relationship between temperature and aerobic scope and to account for inter-individual variability, as this was a repeated measures experiment. So the results of the study show that the aerobic scope of the bronze bream increased with temperature, displaying a linear trend, and there was great variability in the aerobic scope at the higher temperatures compared to the low temperatures. They also showed that temperature had a significant effect on the aerobic scope of the fish. The performance of bronze bream averaged across the thermal gradient showed that we had 2 low performers, 15 high performers, 2 low performers, 15 intermediate performers, and 3 high performers. But when we looked at the performance of fish under each temperature, we found that when fish were exposed to 10 degrees, majority of fish were low performers. At the higher temperatures, 18 and 22, 15 were intermediate performers, and 16 were intermediate performers. So, but then we only had 3 high performers at 18 and 3 high performers and 2 high performers at 22. So the findings of the study showed that the bronze bream displayed enhanced performance under warmer temperatures compared to cold temperatures. And this is because at low temperatures, metabolism is slowed. Fish enter into metabolic depression as cold temperatures inhibit metabolic processes, and it underlies how fish actually perform. Hence the low aerobic scope that was observed at low temperatures. Further increases in the temperature might lead to most fish reaching their maximum capacity of oxygen uptake as water temperatures further increase, the saturation of oxygen in the water decreases. So as we saw in the low performers. The results also show that populations aren't uniform, as some individuals are more tolerant to thermal variability than others. And in a thermal environment that constantly experiences upwelling events and heat waves, like the South African coastline, the presence of physiological phenotypes or individuals is of great benefit to our aquatic ecosystem and our fisheries, as they contribute to the genetic diversity and trade performance of populations, leading to resilient and persistent communities in the face of climate change. And this improves the functioning of our marine ecosystems and our fisheries. So together, these findings suggest that while populations as a whole may tolerate moderate warming, individual differences in thermal performance are critical. Low-performing individuals are likely to be more vulnerable, and whilst intermediate and high-performing individuals may help populations buffer against climate-mediated stresses, making them more resilient against climate change. This is a win-lose for fisheries, as high-performing individuals are able to perform like across a wide thermal gradient because they're more bold, more active, and are more curious. So they tend to be the ones that reach for bait, which means that they are caught easily and catches are easy. But then with time, they will be removed from populations, leaving behind low-performing individuals that are shy and timid and don't really reach for bait and are sensitive to increases and basically fluctuations in temperatures, which then brings us to the importance of spatial planning and management for the purposes of preserving these diverse physiological phenotypes. Because although diversity is thought to exist in exploited populations, studies have shown that protected populations have greater variability in activity patterns and harbor more diverse individuals compared to exploited populations. A spillover from MPAs benefit adjacent fisheries, restoring and replenishing populations and enhancing their resilience. Xolani's talk that's coming up is going to talk more on this. Basically, the impact of MPAs. And so results from the study will be used to, for like future research, results from the study will be used to link fish physiological threshold, like the aerobic scope and phenotypic variability that was observed, to define fundamental niche of the species and to determine how rising temperatures and declining oxygen might constrain future distribution. Sam's talk that's coming up is also going to talk about using such physiological thresholds to predict the distribution of fish. So stay tuned for that. So I'd like to thank everyone who contributed to the success of this project. And I'd also like to thank you for your time and attention. But we did see that, but we did, so the reason why we even used 18 and 22 and 10 in the first place was because of the in situ temperatures that was recorded in this area. But the species is distributed from False Bay all the way to Durban. So there we have other fish, same species, but that are more adapted to warmer temperatures. So the reason for that, we kind of attributed it to maybe long distance dispersal. Maybe some of the fish migrated to come and join this population, making it more resilient. So maybe when we fished fish that are more adapted. So even though they experience these thermal conditions where they are, but they're more adapted to other conditions. Yeah.
Now we come to the audience. Questions? Backward.
A follow-up on that question. Have you looked at the phenotypic variability of a range of individuals throughout their distribution? So fish from Durban against fish from False Bay?
No, we haven't, but that would be a good thing to do because initially we wanted to compare fish from an exploited site, which is what we did here, with fish from the unexploited site, but also considering fish from Durban, that might be a good thing to do for our research. Yeah.
Steve's question about the one that looked like it was dead,
Yeah.
actually.
Yeah.
30 degrees.
Yeah, actually too. Yeah. But that's a good idea. Thank you.
All right. We have to clap again to keep things moving. Thank you very much.